So now we're going to go to Jakub Baczyński, who's going to talk to us about secondary heads.
All right, all right. Sure. All right. Oh, all right. Do you see my presentation?
Yes.
It looks fine. Okay, because I have two monitors and it's sometimes difficult to. Okay, so hello everyone. I'm Jacob, and last week I have officially finished my postdoctoral position at the University of Georgia, where I worked in this large collaborative project mentioned by Paula that focused on understanding the genetic and developmental basis of capitulum evolution. But for quite a while, I've been interested in the evolution of secondary heads or Syncephalia in Asteraceae. And I'm also pretty excited because a few months ago I've received my first serious grant that aims at understanding the evolutionary development of these structures. And during this talk, I would like to present some of my preliminary results, more of future plans, and also possibly hunt for PhD students that might be interested in joining my lab in Warsaw, Poland, because it's more establishing lab right now, but I hope it's gonna be fine. Okay, so to talk about Syncephalia or secondary heads, I think it is good to first establish some operational definition of what I'm going to talk about, and to do that, I took some glossary of morphological terms from the book Systematics, Evolution, and Biogeography of Compositae, and we indeed have some some morphological terms over them, such as secondary heads, syncephaly, as well as glomerulus. And as we can see, syncephalia, or the phenomenon of syncephaly, can be described as a combination of several heads into a new morphological entity resembling a single head. So we have multiple, usually reduced heads combined into a single head-like blossom. And some other definitions also emphasize the presence of a common receptacle or a secondary receptacle in such blossoms. And while focusing on these specific architectures, I am discarding glomerulus a little bit. It's not that I have something against them personally, but this definition as small, compact, irregular clusters of heads, it's a little blurry. And I guess that across Asteraceae, we have many plants that have similar architecture, and it will be very difficult to tell anything more about glomerulus as such. So I'll be focusing mostly on these highly derived forms of secondary heads. So the first thing that's worth discussing is the phylogenetic diversity of secondary heads in Asteraceae, because as far as I know, there hasn't been any work that focused on secondary heads as a phenomenon across all Asteraceae. There are some isolated publications that deal with specific lineages, specific tribes. But based on some preliminary data gathered by Jen Mandel's lab, uh, they found that the structures that can be described as Syncephalia evolved independently in at least 9 tribes of Compositae, and they are present in almost all major subfamilies, including Mutesioideae, so the subfamily that includes Gerbera, but generally some pretty early diverging Asteraceae. And we have a huge morphological diversity of these structures, as you can see, judging by these photographs. But what's most important message to take home is that there are multiple instances of independent origin of secondary heads in Asteraceae, and generally Asteraceae are pretty good at reinventing the heads over and over whenever they have opportunity to do so, which I think is pretty cool. But talking about the evolution of Syncephalia and some hypotheses on how these structures originate during the diversification of the family, I think there hasn't been also much work done, especially not when we compare it to the studies on evolution of the capitulum itself. But some well-established hypotheses from 20th century postulate that secondary heads or Syncephalia evolved by this gradual step-by-step transition. So we have some capitulescence, or branched reproductive shoot that bears capitula instead of solitary flowers. And then this capitulescence undergoes condensation, followed by formation of secondary receptacle and structures called pseudo-involucre in the most derived forms of secondary heads. So to say that the hypothesis to some extent postulates that the evolution of secondary heads recapitulates the pattern that has been described for capitula, but on the higher level of organization. And indeed, when we start analyzing some very complex examples of secondary heads, we can see that there is definitely something going on about this condensation at least. And I would like to briefly discuss some examples, two examples based on the great study of by Regine Klassenbockhoff. It's currently ongoing research effort to understand the development of these structures. And this photo here shows one of my favorite plants, I think, at all. So this is the Polycalyma stewarti from the tribe Gnaphaliae. Gnaphaliae are, in general, they have lots of Syncephalus representatives, and this species is particularly complex because when we take a look on it from afar, it looks kind of like an ordinary daisy, but this external simplicity hides extremely complex blossom, because this is clearly a syncephalium, not a single capitulum. And in this case, we have inflorescence of capitula, a cymoid. So we have a blossom that's dominated by this huge terminal capitulum, and usually 8 cymose units that develop subsequent to this terminal capitulum, and those cymose units produce their own capitula, and this entire condensed structure creates this head-like blossom, and the structure, these perianth-like structures which resemble ray flowers are in fact these membraceous bracts. And so, as I said before, asters are very good at reinventing heads all over, and they sometimes go very far with the degree of modification of their basic body plan to achieve this kind of architecture. The other interesting example is Flaveria trinervia from the tribe Tagidi. And this Syncephalium in this species is very complex. It's not so head-like as in the case of Polycalyma, but it's also quite difficult to say what's going on. But when we take a look on the development, we can understand that this is in fact like a flowering shoot system. We see that there is this terminal aggregate of heads located in the center of this condensed structure, and it is supported by proximal and distal partial inflorescences that are partial capitula bearing inflorescences located below that. And in this case, we see a phenomenon of syndesmy, so incorporation of partial inflorescences and their axis into the main shoot axis. So as I said, the condensation of some branched capitulum bearing structure is definitely one of the ways in which secondary heads of Syncephalia can evolve. There is no doubt that such evolutionary transition happened multiple times during evolution of Asteraceae. But there is also another type of Syncephalia in which I am particularly interested in, and they can be best exemplified by these two species. One is Pycnoceros globosus, like billy buttons. They are commonly sold as Craspidia globosa, which is old name or synonym of this species. So this is this yellow ball on the stick, and this violet ball on the sticks is representative of the genus Echinops from the tribe Cardui. But for today, I would like to focus mostly on Pycnoceros globosus, because this is the species I've been working with during my postdoctoral appointment, and I have most developmental data for this particular species. So why I think another type of secondary heads is particularly interesting, because when we take a closer look on how these Syncephalia develop, we see that especially in the early stages of development, their ontogeny closely resembles in what we see in simple capitula. So we have like the small vegetative meristem that during reproductive transition undergoes this dramatic expansion, and then it starts initiating sub-meristems in these nice contact parastiches. And especially in these early developmental stages, it is virtually impossible to tell this Syncephalia apart from simple capitula. But after the formation of submeristems, they do not develop into solitary floral meristems, but instead they produce small capitula on their own, creating this highly complex fractal-like blossom. And also this Syncephalia also usually bear these expanded receptacles. So those are these structures we would describe as the most derived form of secondary heads. So based on this data, I'm currently hypothesizing, and this is the hypothesis I would like to investigate with in this grant, is that that some types of Syncephalia do not evolve from this gradual step-by-step progression, so the condensation and reduction, but instead they can also develop through a hypermorphosis of capitulum meristem or the floral unit meristem. So this capitulum meristem, instead of producing floral meristems directly, it produces submeristems that repeat the development of the capitulum and only then start initiating floral meristems. And to do that, as I mentioned before, I focused on Pycnosaurus globosus, and firstly I generated the de novo transcriptomes from 5 key developmental stages from this species. So from the vegetative meristem, the primary head meristem, the stage of the secondary head formation stage, the stage when floral meristems are initiated, and lastly the latest developmental stage in which individual flowers start to initiate floral organs. And here on your right side, you can see the PCA on the replicates of these transcriptomic data sets, and we can see that reproductive stages are well separated from the vegetative ones, and that we can see kind of like a developmental progression or trajectory based on these transcriptomes. So my staging was pretty successful, and I am happy because I was very stressed when receiving this data. If everything went fine, as many things can go wrong with this kind of staging. But I have this primary data set upon which I can build some initial analysis and start identifying candidate genes for downstream analysis. So the first thing I have done is simple differential gene expression analysis for comparison between adjacent stages of this developmental progression. And there are some interesting patterns that I have found also in similar data sets for other species and other inflorescence architectures. So firstly, that the vegetative to reproductive transition coincides with dramatic downregulation of genes involved in photosynthesis, and also response to abiotic stresses. But also in case of Pycnosaurus globosus, we can see upregulation of genes enriched in functional terms related to mitotic cell cycle, meristem development, cell division, and also sexual reproduction, of course, because we are transitioning to a reproductive stage of development, which is pretty interesting because it confirms that there is something going on with this initial expansion of this primary head meristem, which also suggests its similarity to individual capitulum. During transition from head to secondary head meristems, there is no enrichment in any interesting developmental processes apart from maybe response to jasmonic acid. But throughout the progression from heads to secondary head meristems and then initiation of floral meristems, we can see gradual decrease in mRNA transcription, and the formation of floral meristems also starts with the switch from proliferation of stem cells to more morphogenesis related processes, because flowers start to develop, which is also expected results, and this the developmental pattern we should probably expect from this kind of morphological trajectory. And also what was quite interesting is that I found no significant enrichment in any terms during transition to floral organ formation. There are also despite that many genes are both up and down regulated, maybe it's the result, it stems from the way that I had to rely on like translating the gene enrichment analysis based on TER, so Arabidopsis database. Maybe those are results of that, but I have to dig a little deeper into this data set to make sure what's going on in here. The other analysis I'm currently focusing on is building a gene co-expression network to identify like co-expression modules which are associated with like the particular developmental stage. And this is also like a data set I'm currently analyzing in order to search for some candidate genes for downstream analysis. And for example, I was able to identify the module called BISC4, which is significantly positively correlated with formation of primary head meristem. And this module is also significantly enriched in terms related to meristem development, regulation of cell communication, signaling, cell division, regulation of growth. So it also suggests that we have some enrichment of genes possibly involved in primary expansion of the head meristem. The other analysis I was focusing on is identification of clavata-free genes in these transcriptomic data sets. Clavata genes and CLE genes in general are quite difficult to recover from transcriptomes and also to like identify correctly. They are very variable, and apart from this CLE domain, so this 12 amino acid functional domain, they are extremely variable. So even so, normal blasting usually fails to recover the right gene for downstream analysis. So to take a closer look on them, I performed the clustering based on a pretty large data set of various angiosperms, and so CLE genes from various angiosperms. And happily, I was able to recover this 1D1 cluster which contains clavata-free orthologs. And indeed, there is a single gene from this Pycnoceros transcriptome. And upon closer investigation, I found that in this functional CLE domain is highly conserved across Asteraceae, including Pycnoceros globosus. So we have this RAAPL amino acid sequence that Paula mentioned in her talk. So at least the clavata-free gene is likely not the main effector that guides this the evolution of Syncephalia in Pycnoceros, but possibly some downstream genes that are affected by, because like as it was already said, clavata-free gene is like a key component of this Wuschel-clavata regulatory loop that controls stem cell proliferation. So there's still a chance that some downstream genes are like these candidate genes for syncephalium development. And having identifying this gene, I was able also to recover the expression of this gene across developmental stages, but I was not able to confirm differential expression between any stages. It's generally expressed at a pretty low level, but still it's quite interesting that interesting candidate gene to look down during subsequent analysis. So for my future plans, and future plans, I would like to firstly focus on assembly of high quality genome from at least one Syncephalus aster. Right now, I am more leaning towards Echinops because the species of the individuals of Pycnoceros I've been growing are likely polyploids with very big genomes. So it's not something I would like to fight with in the years to come. But Echinopses are definitely better. But if some other populations that I have in the greenhouse prove to be diploids, Pycnoceros will be also, of course, a candidate for whole genome assembly. Another part of my grant is focused on comparative transcriptomic analysis between independently evolved Syncephalia, probably between Echinops and Pycnoceros, as well as it's the closest relatives of those two genera that have simple capitula, with the aim to identify the candidate genes for downstream analysis. And by that, I mean mostly analysis of gene expression patterns in situ. And apart from that, I'm also planning to analyze cell division dynamics to confirm like the mantle core histological organization in the primary head meristem, as well as understand some patterning at the cellular level of the Syncephalia. And as I mentioned at the beginning, I'm also currently looking for PhD students. So if you like socialist, modernist architecture, pierogi, bad weather, please feel free to come. I'd be more than happy to have you on board, and I'm looking forward to questions that might be asked. Thank you so much.
That was great. Does anyone have any questions for Jacob? So I actually have, oh, I was gonna say I have, or you can go first, Paula.
Yeah, I was just wondering if anyone has ever tried to make transgenic plants with these species.
No, 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 no. But it's like this absolute. There are also even no regeneration protocols for them. Nobody has ever tried doing any. That's another thing I'm hoping to do in the future. But I did not include it in the grant application because like I have no experience in this kind of analysis. So I was afraid that if I start writing about that, it may.
But do you know, because echinops is a very common ornamental
Yes, yes,
species
yes, yes, yes,
also.
yes.
If anyone has like even like some kind of working tissue culture method for multiplication method for it.
I think I found one paper on tissue cultures or echinops from India when they like established like the media for to like establish Kali from I guess those were like hippocotyls. But I haven't tried this, and I think it was a different species. I think because I'm initially planning to work on Echinops retro, and I think it was done for Echinops spherocephalus. So I'm not sure how easily it would translate to a different species from the same genus. But yeah, there's not so many data on like tissue cultures and no transformation effort. There's like no genome for any of those species right now, and even close relatives. Like I think there's no Gnaphaliae genome as of now. It's another thing that yeah, but.
Yeah, thanks.
So I have like a more systematics question. So you talked about the independent origins of Syncephalia across Asteraceae, but you also said that some tribes also have multiple species that have these secondary heads. Are those also independent origins within the tribe, or is it typically like one origin and then like that, like clade has all secondary heads?
Like at least when I took a look on Gnaphaliae, and because this is like the one of those tribes that has like at least 14 genera, I guess, with some kind of secondary heads, and that those are multiple independent instances of evolution. Like for example, like Pycnosaurus and Craspedia are isolated lineage that are sister to species with generally like simple heads or some like quite complex capitulescences. Yeah, but definitely even within like clades with multiple instances, those are like independent instances of Syncephalia evolution, not like a single clade that. But of course, with some probable exceptions, I guess like with Invernoniae, it's like Lycnophorinae, this the subtribe. This is probably like most of the species in here have secondary heads, but I might be wrong as well because like the taxonomy of Asteraceae is complex. Tagiti too, yeah. The Daniel asked if Tagiti too. I think the Tagiti as well. It's like completely independent, and it's just like Flaviria, and I'm not sure if other species as well.
Any other questions for Jacob or Paula? Okay. Well, thank you both so much for your great talks. We do have a TICA Talks in October as well, and so I hope to see you guys then.
Thank you.
Thank you.
Thank you. Bye bye.